I think CAR T cells is somehow a breakthrough in the treatment of hematologic malignancies. They have really brought us a significant improvement in tumor control and also survival of our patients. The primary indications are for ALL and non-Hodgkin's lymphoma, DLBCL. But we have learned that there are certain peculiarities that we have to address when we are identifying the optimal patients and when we are trying to prepare the patients for the treatment. So what we have learned, Munich is one of the first centers in Europe that has been accredited for both approved products, and so there's quite a learning curve that we are currently going through, and it is still a challenge to get approval by the health maintenance organizations for the reimbursement, because it's a high price, between 300 and 400,000 euros. But we are learning how to address the questions and are actually making some headway here. The next point is to prepare the patient for the leukapheresis. So there are certain drugs that cannot be given while getting ready for the leukapheresis. And then once we have a product that is going into production, not always this will succeed. So sometimes we have to go for a second round. This is something which has been described in the trials, and we also see this in reality. But it's something we are learning how to address. And then probably one of the most challenging aspects is to have a heavily pretreated lymphoma patient that is in an advanced stage. And this patient we need to prepare for the CAR T cell treatment. So we have to invent some kind of a bridging therapy in somebody who has seen many different chemotherapies. So that's not trivial, and it's something we are learning and which we are constantly improving. But it's not always successful, obviously, because we have to bridge a time depending on the production of, for example, 4 weeks after leukapheresis, and that's certainly for a fast-growing tumor a long time. And once we are giving the CAR T cells, then we do see very typical toxicities. The 3 most frequent toxicities are cytokine storm or cytokine release syndrome, neurotoxicity, and fever and neutropenia. And for the cytokine release syndrome, we have learned that tocilizumab, which is an IL-6 receptor antibody, is a very potent drug in controlling this disease early on, or maybe even for prophylaxis. And people have the trend to give this drug much earlier than in the early trials, because we know it will most likely not abrogate the T cell potency against the tumor, but you are much more successful to control the side effects if you give it at an early time point. We also have steroids, which can be useful in advanced degrees of CRS, but also neurotoxicity. And we have the problem, we cannot safely differentiate between a CRS and a fever or sepsis and neutropenia. So we're actually treating both at the same time. And so we're giving antibiotics on the one hand and tocilizumab on the other. This seems to work, although these 2 diseases and the algorithms haven't really been aligned so far, but it seems to be possible to bring these 2 ends together.